In this video, I'm going to talk about the basics of chromatography, the different types of chromatography, and how it can be useful in forensics. Chromatography is a chemistry technique that can be used to separate mixtures into their individual components. So it's very helpful if you have a mixture of, say, colors. You can use chromatography to separate out the different colors in that mixture. So if you look at the picture here, you can see this mixture has a bunch of different components to it. And by using chromatography, you can separate the mixture into its individual parts. And then you could analyze the parts, purify them, so on. So it's very useful here in being able to separate mixtures of substances. Since chromatography is a chemistry technique, it's important here that we review some of these chemistry terms. So a mixture is when you have two or more substances combined, but they're not chemically combined. So they're only a physical blend, so they can be separated here by physical means. So like air is a mixture of different compounds, a bowl of cereal, Kool-Aid, these things can all be separated. But a compound is when you have two or more elements that are chemically combined. So you cannot separate a compound without it undergoing a chemical change or a chemical reaction. So salt is NaCl. It's sodium and chlorine together. They are chemically combined. Water, carbon dioxide, these are chemically combined and you cannot get them apart by physical means. They would have to undergo a chemical reaction in order to come apart. Some more chemistry terms here that are important to understand. A solution is a mixture, okay? It's when you have one substance dissolved in another. So a solution can be separated by physical means, all right? The two parts of a solution are the solute and the solvent. The solute is the substance that is dissolved in a solution, and the solvent does the dissolving. Okay, you might have heard water called the universal solvent because it can dissolve a lot of substances. So if you look here at the table, you can see some examples. All right. So if you, for example, if you looked at lemonade, there's probably a lot of different solutes here that could be in lemonade, but the main solute in lemonade would be sugar. Sugar is dissolved by the solvent, which is typically the juice or the water. Okay. If something is insoluble, it does not dissolve in water. And if something is soluble, then it dissolves. Okay, not everything dissolves in water. Sand is insoluble. It does not dissolve in water, but salt is soluble. It would dissolve in water. Same thing for sugar. It's soluble. It would dissolve in water. Chromatography has a lot of different uses, okay? You can use it to separate out the inks and dyes here in different substances. So for example, you could use this in pens or markers or even, different colored candies. You can actually use chromatography to see the different colors that might be present. You can use this to separate out the pigments in plant leaves. It can be used to determine the chemical composition of different substances. And in forensics, we use it in a lot of crimes that might involve like ransom notes or forgeries because of the fact that you can use it to separate out different pigments. You might be able to figure out, which pen was used or, which marker was used in the writing of the ransom note or the forgery. So it definitely has a lot of uses. There's a lot of other uses for chromatography, including being used by pharmaceutical companies to determine the amount of chemicals in a new product. It could be used to determine the alcohol level in a person's bloodstream. You could even use it to figure out the level of pollutants in a particular water sample. It's also useful in purifying substances, maybe in the manufacturing industry. So there's an example of chromatography over here on the left. You can see this sample here, this black sample is being dumped through the tube. And basically, as it goes through the tube, certain colors will come out faster. So it looks like the yellow comes out first. So you could gather all the yellow. The red would come out second. So then you can gather all the red. And then the blue would come out last. So that basically, as it traveled through the tube, separated out the different colors. Okay, so there's definitely a lot of uses here of chromatography. So how does chromatography actually work? Well, there's two phases involved with chromatography. The mobile phase is the part of the chromatography that usually involves like a liquid moving. There's some form of solvent here that will move through the apparatus or move through the paper. that will bring the substances with it. And then there's a stationary phase, which is the part of the apparatus that does not move. If you were doing this on paper, the stationary phase would be like the paper. The paper itself is not moving, but the liquid or the solvent you're using would be moving, okay? So basically, the substances are either going to be attracted to the liquid and will move, or they're going to be attracted to the stationary phase or like the paper and not move. So if a substance has more of an attraction to the solvent or the liquid, it's going to move farther. Okay, it's going to travel with the solvent and it's going to move up the, in this case, paper a lot further. If a substance is more attracted to the stationary phase or the material, it's going to not move as much. Okay, so it all depends here how attracted a particular substance is to the mobile phase or the stationary phase. If you're looking at the example over here on the left, all right, if you were starting here at the bottom and your liquid was traveling up the paper, and we're going to talk about this a little bit more, but that would be the liquid itself moving up your paper, the solvent moving up the paper would be your mobile phase, all right, and the paper itself would be the stationary phase. So if you look at these colors here, the blue is closer to the bottom, so it's going to be more attracted to the paper, and then the yellow was closer to the top, so it's more attracted to the liquid or the solvent, and it's moving with it. Okay, and the red's kind of in between. So this is just a quick overview of how this would work. All right, you have some type of mixture that you might put on your chromatography paper. There are different forms of chromatography, but if we were doing this in lab, we'd be using paper. All right, so your mixture would be placed on the paper and you'd have some form of liquid here that you put the paper in. So as the liquid moves up the paper, the different components of the mixture separate out based on their attraction to the paper or the liquid. So in this case, it looks like the yellow was more attracted to the liquid, so it moved with the liquid, whereas the blue was more attracted to the paper, so it stayed behind. So this is just kind of a little bit of an overview of how this would work. There are different types of chromatography, and some are more common than others in a high school lab or a college lab. But there's something called liquid or column chromatography that uses this column to separate the substances. Basically, you dump your sample through it, and based on the attraction to the liquid or the material, the substances will come out the bottom of the column at different times. There's something called TLC or thin layer chromatography, which basically uses these liquid samples that are placed on the slide and they will move through it. So usually it's a glass plate that has this thin layer of alumina or silica gel. So you plate your samples on there, let them dry, and they will move through the plate differently. There's also gas chromatography, which would basically kind of analyze these gas samples with a computer, and you can look at a graph to determine the different substances. But then the type that's usually most common that we would use in lab is paper chromatography. So you put these dried liquid samples on the bottom of the paper, and then you run the solvent through it. And as the solvent moves up, as we saw, the different substances in your mixture would separate out and you could see them differently on the paper. So if you look over here on the right, this is showing you some paper chromatography with some different plant pigments. So they spotted the plant pigment here at the bottom and they allowed the solvent, the liquid, to move up. And as it moved up, you can see the different colors here separating out. So that's an example of paper chromatography. So the basic setup of paper chromatography, you take some form of absorbent paper. You could do this with filter paper or coffee filters. You would draw lines near the top and the bottom. So the bottom line is where you would put your samples, and then the top line is where you would want to stop it. When your solvent got to that level, you would want to stop the chromatography. So whatever material you're looking at, your ink or your mixture, you would dot them on the bottom, okay, on the bottom line, and you try to keep them spread out. Okay, you don't want your samples below that line, because when you put your paper in the liquid, if they were below that line, they would just wash off. And then usually what you do is you suspend this paper in some way in your liquid and you allow the liquid to run up through the paper, but you need to stop it once that liquid reaches that top line. And then you remove the paper, you can mark how far up the liquid went, and you can mark the colors, and then you can analyze them to determine if any of them were mixtures or if you were testing a mixture to see which different substances were in that mixture. This is just an example of a chromatography setup. It looks like we have our chromatography paper here being placed inside maybe a beaker. The liquid level is in there. You can use different solvents. Sometimes it's just water with maybe a little bit of salt in it. Sometimes you might want to use like rubbing alcohol here. There's a lot of different solvents you could use. But notice there is an ending line here at the top and a starting line at the bottom. And the samples were all placed on the starting line here at the bottom. And then you suspend that paper in the liquid, but notice you don't let the liquid go above the colors, otherwise they would wash away. And soon as you put this in the liquid, all right, the liquid is going to start moving up through the paper. This is an absorbent paper, so like a paper towel, you would expect the liquid to move through it. And as the liquid kind of travels up through the paper, these colors will move along with that based on their attraction either to the liquid, the mobile phase, or they wouldn't move as far if they were more attracted to the paper or the stationary phase. Okay, so you would run this up. Once your liquid level got to this top line, you would pull it, you would mark your liquid level, and then you would analyze the different colors and see how they move. Part of the analysis of the colors is looking at the RF factor or the retention factor. This can basically be used to identify a particular color, and you could compare different spots to see if they are the same substance. Basically, you would take it's kind of a little math problem. You would take the measurement from the distance of the origin, which would be the bottom line, to the spot itself, and you would divide it by the distance from the origin, the bottom line, all the way up to the solvent, how far the solvent ran. So this is why marking these things would be important, but you just use a ruler and you would measure these things. Okay, so if you were looking at it over here on this particular example, okay, if your initial spots were down here, that would be your origin. Okay, if the solvent ran to up here, that would be your solvent front. All right, so you would measure from your origin to where the spot went, usually the center of the spot, and then you would divide that by the distance from your origin all the way up to your solvent line. Okay, typically here, if two substances have the same RF value, they're usually the same substance. Okay, so this would be something you would analyze once you did the chromatography. You would make these measurements and be able to hopefully compare and identify substances.